Okay, my name is Dr. Jerry McLaughlin, and I'm an emeritus professor of pharmacology from Purdue University in the pharmacy school there. And I'm going to talk about Paw Paw and cancer from discovery to clinical trials. This will talk a little bit about some of my life's work. First, some United States cancer facts and figures for 2005. You can see that these are listed there. It's important to note that there are almost 600,000 deaths per year due to cancer. To put that in perspective, there were like 600,000 total deaths in the Civil War, in the American Civil War, back in the 1860s. And that was in 4 years. So here we have as many people dying each year of cancer as died in the Civil War. Further perspective, 60,000 deaths, about 59,000 or 60,000 deaths in the Vietnamese War and all the years of that we had for American casualties. So each year we killed 10 times more people with cancer than we did in all the Vietnam days for that war. So this takes a terrible toll on our society, and it's not just in lives, but it's in money. And the total costs of cancer to the American healthcare budget are someplace in the neighborhood of $189 to $200 billion per year. And one man in 2 can look forward to getting cancer sometime during their lifetime, and 1 woman in 3, and 1 woman in 8 will get breast cancer. And we know that this is going to happen unless something dramatically happens in the future. How can we fight cancer? The typical cancer treatments are chemotherapy, radiation, and surgery. And let me review those briefly. Let's take radiation first. Ionizing radiation can go through cells from gamma sources or cobalt sources and ionize the cellular components so that they scramble substances such as the DNA, and it becomes impossible for it to function. Radiation works just fine, except that in order to get to the bad cells, you have to go through some good cells. And the collateral damage to the good cells causes all kinds of problems and can even be fatal in cases like brain cancer. Surgery. Surgery works if you can completely remove the cancer. Surgery can be curative. However, cancers spread very quickly, and cells broken loose by cutting through tumors can end up in the lymph system and spread the cancer that way. I'm going to go for it. Attack cancer is chemotherapy. And as a consequence of chemical warfare, our discovery of nitrogen mustard led to the first chemotherapeutic agent. So, as a toxic material, we found out a way to treat cancer. And nitrogen mustard, when given to cancer, will cross-link the DNA structures. Remember that the DNA is a double-stranded helix, 2 strands of the DNA. You cross-link them chemically and they can't separate. And so the cancer cell cannot replicate its DNA because it's cross-linked, and the cancer will then, the cell will die. It also alkylates other cells, good cells. And I should point out that chemotherapy, people who survive chemotherapy, 10 to 15% of those people will downstream from that time develop tumors due to the chemo, because most of the chemotherapeutic agents are mutagenic. They're cancer-causing chemicals in their own right. So nitrogen mustard is a very potent mutagen. Antimetabolites are another class of chemotherapeutic agents. These include compounds like methotrexate, which is a fake folic acid. It looks like folic acid. I mean, the layman looking at the structure would say it's folic acid, but it's not. It's different than folic acid. And so it goes into cells like folic acid, is taken up by the cell, and then inhibits the methylation process for making T, which is thymine for the TTP, thymidyllic acid we call it. And you have to methylate, and the methyl group cannot get transferred in the presence of methyltrexate. Other newer components of chemotherapy include the tubulin inhibitors, and these include the vinca alkaloids, vincristine and vinblastine, from the periwinkle plant discovered at Eli Lilly by a friend of mine, it's Gordon Svoboda. And these agents will cause a polymerization of tubulin, which is an elastic-like protein that pulls the chromosomes apart during cell division, and then a new cell wall will form down between. And when those chromosomes, this elastic protein tries to pull them apart, but it gets inactivated by the vinca alkaloids, and this is the phase of cell division called metaphase, and so the cells will sit there in what we call metaphase arrest, and they're all just sitting there with their chromosomes ready to come apart, and it'd be pulled apart, and it doesn't work. So topoisomerase inhibitors include some things that there's an enzyme that goes along and repairs our DNA and links it back together again. It's called topoisomerase, and because it goes over the topography of the DNA strand, and there are compounds that inhibit that and prevent the DNA from repairing itself, and thus they're anti-cancer agents. So unfortunately, we don't have a lot of new ideas for mode of action of anti-cancer agents. We need some more new ideas. So those are the three things that the physician has: surgery, radiation, and chemotherapy. I should also point out the side effects of chemo. In addition to causing cancer, chemotherapy causes hair loss, GI bleeding, bone marrow depression, which of course results in infections, thymocyte depletion, which are platelet depletions, which means that your blood can't clot properly. You have a pink toothbrush for a while. And red cell depletion. So erythropoietin has been developed in order to stimulate your bone marrow to make more red blood cells. And almost every night on TV you'll see somebody saying that they were so tired until they took erythropoietin and now they can go play with their grandchildren again because of the chemo. So actually what's happened now is that these drugs used to treat the side effects of chemo have become some of the top 10 selling prescription drugs in the United States. So it's become a big business to treat the side effects of chemotherapy, let alone the chemotherapy itself. It's important to keep down infections once your white cells are depressed, from the bone marrow depression. So, and they're actually, for thrombocytes, actually you can get other people's platelets now. And so some people have to take other people's blood platelets, and they combine the blood platelets from 10 different patients and inject those platelets into the cancer patient. And then what happens is they have allergies to other people's platelets, just like you can't take somebody else's blood type. And so you develop your hives and immunity against the other people's platelets, and so they give you Benadryl and antihistamines, and they have all sorts of ways to get around these problems. I began working at Purdue University in 1971, and in 1973 or 72, President Nixon declared war on cancer, and so there became money available for grants for professors to work on cancer and cancer treatments. So I, in 1973, got my first little grant. It was a grant to look at cactus plants as a source of anti-cancer agents. And I got that actually through the American Cancer Society. I believe it was LaGrange County, Indiana Cancer Society gave some money through Purdue's Cancer Research Center, which we had established, or were establishing at that time. And that's how I got started. Dr. Chang and Dr. Cassidy and I, by 1976, had qualified and competed and earned a contract from the National Cancer Institute to fractionate plant extracts to try and identify the compounds in plants that might be useful as new anti-cancer agents. And we had those contracts for a total of 6 years and went through literally hundreds of species of plants that were being shipped to us from all over the world, looking for anti-cancer activity. I then was able to get support from the National Cancer Institute for R01 grants for a total of 13 years. And R01 grants are the hardest grants to get because these are the researcher-originated grants. In other words, you got to have a new idea and go in and compete with everybody else's new idea and try to get funding. And I was able to get that type of funding for 13 years or so. So in my 13 years then of work, I went through over 3,500 species of plants, getting those plants in from various sources and going through them one by one. Now, I used a biological assay called the brine shrimp test, and we developed that brine shrimp test in my laboratory so that we could rapidly screen plant extracts looking for things that might kill cancer cells, because all anti-cancer agents that I tested killed brine shrimp. So I thought, well, anything that kills brine shrimp might end up being an anti-cancer agent. And that was a real godsend, because the cancer, normally you screen against cancer cells growing in culture, and that's at the cell culture lab at Purdue. That assay cost us $120 to assay against 6 different types of human tumors. I would have soon used up all my money on the bioassays if I hadn't developed the brine shrimp test. And to follow that up, we developed an assay using potatoes and growing tumors on potatoes called the Agrobacterium tumefaciens bacteria. Has a plasmid that it induces into a plant cell, a wounded plant cell, and in about 14 days we're able to count 30 little tumors on a little round disc of potato. And anti-cancer agents will stop the development of those tumors or make a significant decrease in the number of tumors. And so here we had, using potatoes and shrimp, I was able to assay for anti-cancer activity. So we then proceeded and went through these plants that were positive and ended up isolating over 350 new compounds that have anti-cancer activity. Now 200 of those compounds approximately were a new class of compounds called the Annonaceous acetogenins, which we got into by looking at the Paw Paw, which we first found activity in 1976. Here we have a picture of Paw Paw, which are called the Indiana banana. The ripe fruits of these taste like banana custard, and they've been eaten by Native Americans and pioneers and the Lewis and Clark expedition explorers and all kinds of people as a fruit that is quite sustaining. It has a lot of protein, and fats, and carbohydrates in it. And also, it has these compounds called Annonaceous acetogenins. And some workers at Kentucky State University have figured out that even the ripe pawpaws now have the acetogenins in them, which was a little bit of a surprise. There are certain varieties that don't, but some of the varieties do have actually amounts of acetogenins that are equivalent to that of the twigs, which are the plant part that we typically use. The scientific name of Paw Paw is Asimina triloba. It's in the Annonaceae family. And it's interesting that a fluid extract of the seeds of Paw Paw was sold as an emetic way back at the turn of the last century, 1898, by Eli Lilly and Company. An emetic is a product that makes you vomit. So Paw Paw will make you vomit, and indeed that's the side effect that prevents people from overdosing. If we see this next slide of the United States shows the eastern states, and the darkened area shows where Paw Paw is native. And it's a native of the eastern United States, from eastern Kansas east on to the Atlantic seaboard, and from Michigan, central part of southern Michigan, down to the state of Florida, and all the way to the Gulf Coast and Mississippi and Alabama. The family Annonaceae is quite a large family. It's 120 genera, about 21 or 2,300 species in it. They're mostly trees and shrubs, and they're all, except for Paw Paw, tropical or subtropical. Our next slide here shows us a picture of guanabana, or Annona muricata. It's also called graviola, or some people have called it Brazilian Paw Paw now, trying to cash in on the good name of Paw Paw with this product. we've looked at several types of these graviola capsules that are currently being sold, and there are a couple of them. One of them was 24 times less potent than the Paw Paw product that's being sold, and another one is 56 times less potent than the Paw Paw product. So those products essentially are worthless if people are taking those for cancer, because there's two strikes that they have against them. Number one is that they have the wrong structures of compounds in. They have the single ring compounds instead of the double ring compounds. And the second thing is that they're not standardized biologically, so that nobody knows what the potency is. If they might be a good batch or a bad batch from one bottle to the next. And so you don't know actually what you're getting. The next slide shows us these Annonaceous acetogenins. This is a rapidly growing class of natural products. There's probably over 400 that have been isolated now since 1982, when the first one was found and published by Dr. Jack Cole's group at the University of Arizona. They're only found in several genera of the Annonaceae family. They have very potent in vivo anti-tumor effects. They're quite cytotoxic. They're pesticidal as well. They kill insects. They kill bacteria and fungi, and we know that they're antiviral as well. They're derivatives of C35 or C37 long-chain fatty acids, and they're end with a terminal methyl group that's methylated on alpha-beta unsaturated gamma-lactone, or else they flip over and form a ketolactone, and sometimes they have none, or sometimes they have as many as 3 tetrahydrofuran rings in their structure, and we found some now with tetrahydrofuran rings, which are 6-membered instead of 5-membered rings in the structure. The structure illustrated there is the structure of butylysin, which is a compound that has 2 tetrahydrofuran rings that are not adjacent. They're separated from each other. This next slide shows us some of the diversities of structures, and as I said, we found over 200 of these. And incidentally, we've done some really beautiful chemistry in figuring out these structures and figuring out their 3-dimensional positionings. And that work, I think, is what I'm most proud of, all that chemical work. And we've published it in journals like Tetrahedron and Journal of Organic Chemistry and the Journal of the American Chemical Society and Journal of Natural Products and other journals, Phytochemistry, for example. The top compound we see here is butylysin. We originally got it from Annona bullata, but it's one of the major components of Paw Paw as well. You see there the 2 tetrahydrofuran rings in the middle are adjacent to each other, with this terminal lactone ring at the other end. And you see from these other structures the diversities that we can have with single ring compounds or double ring compounds or non-adjacent, and even a 3-ring circus, as we call it. I kept telling the research group there's got to be one of these compounds with 3 rings in the structure, and sure enough, Dr. Gu found one. He was one of my postdocs, and found one in Goniothalmus giganteus. Unfortunately, with 3 rings in the structure, the biological activity decreases and doesn't go up. The optimum structure are those compounds that have 2 rings that are adjacent to each other, with the ring system starting at carbon 15. And we've done structure-activity relationships in cell culture against mosquito larvae and in isolated mitochondria, and of course in brine shrimp. And we can definitely say and point out what are the most potent compounds. This next slide shows the stereochemical aspects of the major Paw Paw acetogenins. You see those letters from right to left going A, B, C, and D, and E. And we see that each of those points out an area of chirality where there's a potential of having more than one structure from the structure that I've illustrated there. And indeed, as you go across those 2 rings, there are like 6 chiral centers across there, and then 2 more chiral centers in the molecule, giving us a total of 8 chiral centers. If we just look at the flat structure there, the number 2 to the 8th comes to mind, then, and that's 256. That means that this flat structure is represented in actuality by 256 different structures. So which structure does nature make? That's what we had to figure out. And we figured out ways to do that. Okay? And we can see here that trilobicin, acimicin, and biotin are three of the main structural types depending on the stereochemistry of those ring systems. This next slide shows us the HPLC chromatogram of just a fraction of a Paw Paw extract, and all those little peaks there represent one or more compounds. You see how complex that extract is. This was done by Dr. Zhao, one of my Chinese graduate students who has his PhD now, and he identified every one of those peaks, isolating the compounds and proving their structure and testing them biologically. Over 50 of these peaks then have been isolated from Paw Paw, one by one, and structurally characterized. The next slide shows a little bit more about this brine shrimp lethality assay, or the brine shrimp test. You probably have heard of these as sea monkeys, and you can buy the eggs of them and put them in a little salt water at the concentration of ocean water, and they'll hatch. And you can raise them, actually. It takes them about a month, and they'll become adults, and they'll actually swim around and reproduce and lay eggs and all that. But we only use them from 48 to 72 hours after hatching, as they're still in the larval stage, because at that stage they're quite susceptible to chemicals in their environment. And so we can kill these little brine shrimp, and since they're invertebrates, nobody cares if we kill them. If they had a spine, then people would care, but nobody cares, at least not yet. So we might have the Brine Shrimp Protective Society formed here any day now. But we can record the number of these that are killed and find the concentration that will kill 50% of them, and this gives us a number that is quite reproducible experimentally and allows us to actually get confidence levels for statistical purposes. Okay, once we got this brine shrimp test, then we can go through the different parts of the Paw Paw tree. Initially we worked on leaves and twigs, a mixture. And so we extracted different parts, and we can find here that the leaves down at the bottom of this slide show us that they're not very potent because it's a bigger number to kill 50% of the shrimp. But the little twigs were, many times more potent than the leaves. And the unripe fruit was potent, and the root wood was potent, and the seeds were quite potent, and the stem bark. But the wood itself, the stem wood, is not very potent. So from this, we'd been previously using the bark of the trees, but we have to kill trees to get the bark. So we decided the twigs make a logical source, because you can just trim the trees and trim off the twigs. And I have another slide, which I'm not going to show here, but that shows us the diameter of the twigs. If they're 1/4 inch and less, really gives us a really excellent potency to the twigs. We also were testing the Paw Paw extracts against pests, and actually Eli Lilly and Company did this for us down at their Greenfield lab, which now I think Dow Chemical controls. And you see that we were able to kill mosquito larvae with an FO2O extract. Down 100% of them died at 10 parts per million. And the guy who did this testing for me called me up and said, "Jerry, what the hell is this stuff?" And I said, "Why is that?" And he says, "Because we're killing mosquito larvae at 10 parts per million, which is more potent than many of the pesticides that these synthetic chemists are giving us." And at that point was 1982, and I didn't know what was in there, and we started working on it feverishly. And by 1985, we had the structure figured out for the first compound. So 3 years of work. You also see that we were killing C. elegans. C. elegans is Caenorhabditis elegans. It's a worm, and it's a typical worm that you would use to test for killing parasites. And so we see we're killing the parasites very well. And so from this, we're able to develop a couple of products, a lice remover shampoo, where we introduced this in September of 2001, and there's a publication there in Phytomedicine where we did a clinical trial with it, and 16 kids, all 16 of them got rid of their lice, and some of the kids had had lice for 3 years. So, and we had some nurses do that study for us, and so we knew we had something there. Nature's Sunshine has parasite cleanse, and so we put Paw Paw capsule into that mixture of product, and that combines it with pumpkin seeds and black walnut hulls and a little cascara sagrada and artemisia and clove and garlic and some other things, and it's quite effective at removing intestinal parasites now. But that gets away from our cancer story. I want to point out here, why do we use the twigs? Well, as we can see from the previous brine shrimp study, it was the most potent plant part, and it's easy to collect, and it allows the tree to survive, which is going to give us a sustainable resource. But I want to point out that this slide illustrates a collection of a bunch of worthless material. And this is because we were collecting it in November. So the time of collection, we figured, now must be important. So we had to do a complete study that took a whole year, determining the biological activity of the Paw Paw each month of the year as collected from the same tree to eliminate any variation in the genetics of the trees. And sure enough, as you can see, the 5th month here is just a big peak in biological activity, and the 5th month is May. So the twigs are most potent in the month of May. So we want to collect it then. And we also, in the same study, tested with HPLC for acimicin and belotocin and trilobicin, and those peaks superimpose over the biological activity peaks, which show that those compounds are highest at that time of year as well, indicating that those compounds are probably responsible for the biological activity. Okay, so got to collect it in May. Another variation that people don't think about very much is the genetics of the tree. Like, which tree is the more potent, this one or that one? So we went out and collected actually 667 twig samples from that many trees, and collected them all in one day. And so then we brought them back to the lab, and we only struggled through 135. It took us a day to assay each one. We went through 135 of these twig samples, getting the brine shrimp activity, and it was really a surprising result. From that plantation of Paw Paws, in the 4th row, the 11th tree in the 4th row gave us a brine shrimp activity of 0.033. But the low producers had a tree that gave us, for example, the 2nd row, the 32nd tree, that tree was only 32. The difference in biological activity between those two trees is 1,000 times, meaning that one tree is 1,000 times more potent than another one. So if you're going to be collecting trees or growing trees, it would be wise to make some cuttings from this more potent tree, right? And so we've got those trees labeled, and we know which ones we can do, and we can clone those trees, do grafts with those trees, and produce some very potent Paw Paws, Paw Paw extracts. Okay, there are 2 sites of action for these Paw Paw compounds, and one of these is in the cell membrane at a system called the NADH oxidase system, and this takes NADH back to NAD and allows us to take glucose down to pyruvic acid through glycolysis biosynthetically and do it like yeast do, anaerobically. So it's pretty obvious that a lot of tumors grow without air, without oxygen. They get very little oxygen. And so they grow and metabolize glucose without using oxygen, like a yeast. And by inhibiting this system, we stop the anaerobic production of cancer cells without air. Now, most cancers, we think, probably are getting air from oxygen dissolved in lymph that are bathing the cells, along with glucose. In that case, they're using oxidative phosphorylation in order to phosphorylate ADP to ATP. ATP is the key energy compound, and that's what we're getting from the energy of glucose, from burning it up to carbon dioxide. And so when we burn it up to carbon dioxide, we're getting ATPs produced. but we work in the mitochondria there, where ATP production takes place by oxidative phosphorylation. And so this ATP is a key compound for energy, and it's also a key building block for the nucleic acids, DNA and RNA. And so the cancer cell can't make new chromosomes without ATP. Okay? And so by depleting it, we're throwing a monkey wrench into the gears of the cancer cell's reproductive machinery, and it can't grow. Two ways that this material works. The next slide illustrates in human leukemic cells that we are able to not only decrease ATP, but we also decrease the other nucleotides as well. And these are the four basic bases that go into making ATP, or that go into making nucleic acids like DNA and RNA. Okay, the A, the G, the U or T, and the C. And by depleting these building blocks, you also stop the cancer cell from growing, or you stop a virus from replicating itself in an infected viral cell, in a viral infected cell. Okay. So there's another way that the material works, and we know that we can treat viruses like shingles and herpes with the Paw Paw material as well. The next slide shows us a little cartoon of the P170 glycoprotein. That's the multi-drug resistant pump. And this is significant because it develops in the cell wall or the cell membrane of the cancer cell that's being treated with chemo. And after this is developed into this cell membrane, that cell is no longer susceptible to the treatment of chemo. And this is why in time chemo no longer works, because this membrane bound protein is there as a little pump that pumps the drug out of the cancer cell. Now, Paw Paw works against these multiple drug-resistant cancer cells that have this pump. I first saw that in 1988 when I saw that we were killing MCF breast cancer cells that were drug-resistant. We were killing them at lower doses than we were killing their parent cells when we used the Paw Paw compounds. And with everything else like adriamycin, it took 1,000 times more to kill the resistant cells. Here we're taking 5 times less to kill the resistant cells, and we couldn't believe it. And I called the National Cancer Institute, Matt Sefness, and said, Matt, we're killing drug-resistant cells. This has got to be significant. And he said, what's the mode of action? Tell me the mode of action. That was 88. It took us until 93 to determine the mode of action that we did it. Now, key here is ATP. ATP, 2 ATP binding sites. Grab onto ATP, the ATP is cleaved to ADP and phosphate. That gives off energy that makes the pump go, and it spits out the drug. It just keeps pumping. Without ATP as the energy compound, the pump can't run. If the pump can't run, cancer drugs should stay in the cancer cell and work, even though the patient has become refractory to chemotherapy. In the presence of Paw Paw, it should deplete ATP and cause chemo to work or to restore the susceptibility. That's important clinically to keep in mind. This next slide just shows us cytotoxicities of several of the acetogenin compounds, as done at Abbott Laboratories by Dr. Jim McAlpine. And this shows that we're out here around 10 to minus 6. That's micrograms per mil. That's down to a millionth of a microgram per mil, which is a very, very small concentration with the 3 major Paw Paw compounds. And the single ring compounds are much less. And adriamycin is the top compound, which is a key anti-cancer drug today. You see it's only effective around 10 to minus 1, 10 to minus 2. The next slide comes to us from Wayne State University. Tom Corbett up there took some of the Paw Paw compounds and ran them against drug-resistant breast cancer cells and found that it killed the drug-resistant cells very well. He also ran adriamycin. It wouldn't work because, of course, it's being pumped out by the pump. And he then ran some normal cells that aren't cancerous and found that adriamycin is more toxic to the normal cells than the Paw Paw compound was, which is good to know. Then we got Upjohn to help us, and over in Kalamazoo, and there they were able to demonstrate against L1210 mouse leukemia that Paw Paw is like 300 times as potent as Taxol in vivo. And at the same time, the Taxol mice lost 10% of their body weight, whereas the Paw Paw compounds actually cause an increase in body weight, showing that the mice are not sick. And the duration of this experiment is 10 days. The next slide shows us another study done at Upjohn Forest, and here they took human ovarian cancer. This is Gilder-Radner cancer, and it was called A2780 human ovarian cancer. And they put it into athymic mice, and these athymic mice are so inbred that they have no thymus gland, and so they cannot reject anything. So you have to grow them in germ-free atmosphere so that they don't get infected with anything. And you feed them sterile water and sterile food, and you glove up and don't breathe on them or anything as you're handling them, and it's pretty expensive to do this. So the information on this one slide cost over $60,000 for the experiments to be done. And you can see here that cisplatin, which is a standard anti-cancer drug for ovarian cancer, caused a 78% tumor growth inhibition in 10 days. But these mice lost, with cisplatin, they lost about 5% of their body weight. And with belotocin up here, we can see that we had about a 67% tumor growth inhibition versus 78% for cisplatin. And belotocin causes 75% tumor growth inhibition, which is pretty close statistically to the 78% of cisplatin. And we had much less weight loss with the Paw Paw compound than cisplatin. Cisplatin's a nasty drug. I've talked to people that have taken it like for disseminated prostate cancer, and they have to be almost roped and drug into the doctor's office to get the next dose. It's extremely toxic and it makes you sick. So those were good results. Now, programmed cell death is a term that you'll hear about when you're dealing with cancer. It's called apoptosis, or programmed cell death. And another type of cell death is necrotic cell death that you'd get like if you burn a cell. The heat would destroy the cell. Okay, if you kill the cell by programmed cell death, in that process there are natural changes that are taking place in the cell that create enzymes called DNAases that break the DNA down into little fragments so that the next cells to come along can actually scavenge some of the cell components, you see, and use up those cell components. And so if you extract those cells, extract the DNA, and run an electrophoretic pattern of the DNA from the necrotic, or from the apoptotic cells, you'll see a laddering. This is like you see after they do some cleavage in the DNA testing they do now to determine paternities and identify people's hair and skin segments and cheek washings and things like that. So you can see here in the center this nice beautiful laddering as caused by Paw Paw. On the right we see a standard DNA sample, and the two blobs at the bottom are necrotic cell deaths. These are cells, identical human leukemic cells. Actually, these are B cells, which are a type of leukemia cells. And there's no cell laddering there because those are necrotic cell deaths. So this is causing programmed cell death, the Paw Paw is. And this is the type of death that you want to have for cancer. All good anti-cancer agents cause this type of cell death. Okay, we've had over 100 scientific papers then on the chemistry and the biology of the Paw Paw compounds, and these are in good journals, nice quality scientific journals. The Journal of Natural Products, which is published jointly by the American Society of Pharmacologically and the American Chemical Society over in Columbus, Ohio, back in 1999. For the first 6 months of that year, ran this picture of Paw Paw and the structure of asamicin on the cover to honor our research. Also on that picture you'll see a butterfly. And that little butterfly, I was just down to my Paw Paw patch yesterday, cutting weeds from them, and I saw one of these butterflies. And they're supposedly an endangered species of butterfly. There's a zebra swallowtail, a black and white butterfly that's striped like a zebra. Okay? And this butterfly is an obligate parasite on Paw Paw. In other words, it lives by eating Paw Paw leaves. And that little butterfly yesterday was busy laying eggs on my Paw Paw plants. But I tolerate a few of them because they're not destroy that many. But the question is, why does this butterfly live on Paw Paw when everything else, other insects, are killed by the Paw Paw acetogenins? So I wanted to know. So I got a guy named, I can't think of his name now. He's at Widener University, and he's a world's expert on zebra swallowtail butterflies. So he got me some zebra swallowtail butterflies and the larvae of these butterflies. We extracted those, freeze-dried the extract. We freeze-dried them first, then extracted them, and then assayed in the brine shrimp test. And guess what? The brine shrimp died. So the butterflies were carrying something that was toxic. So then what could that be? So then we ran HPLC mass spec with the help of Dr. Gu, who's now at Xenobiotics Labs. And I just saw the guy's name, Steve Matigowski at Widener University. And we ran, and we could see then the peaks for the major Paw Paw compounds in the butterfly extract, which means that the butterfly is concentrating the Paw Paw compounds from its diet. And thus, if you're a bird and you eat that butterfly, what's going to happen to you? You're going to throw up, because the Paw Paw compounds are emetic, right? So here's a protection that these butterflies have, and that's why the butterfly can be so beautiful and say, here I am, nice black and white, don't touch me or I'll get you, And so birds leave them alone, and actually leave the larvae alone, because the larvae are very brightly colored as well. So we proved that and published that. Actually, I had an undergraduate do that work, Dr. John Martin. So there are 4 mechanisms by which Paw Paw regulates cells then. We slow down or stop ATP production in cancer cells and deplete the energy, and programmed cell death or apoptosis is a result of this. And we do this by hitting the NADH ubiquinone oxidoreductase enzyme of the mitochondria. This is the PSST protein subunit of complex 1. And then we also do it by hitting the NADH oxidase in cell membranes. And ATP is a direct precursor of DNA and RNA, and we thus stop the production of those viral components as well. Angiogenesis, or developing new blood vessels into a tumor, requires ATP. And by depleting ATP, we deplete angiogenesis. So new blood vessel formation can be stopped by ATP depletion. And the multiple drug-resistant pump, which is the bugaboo of chemotherapy, requires ATP. And we've already demonstrated way back in 1988 that we can stop those cells that have that pump quite effectively with the Paw Paw compounds. And this should allow us to actually combine the Paw Paw compounds with chemo and help restore susceptibility. Then, moving on to vertebrates. And we had to do a dog study in order to try and have people actually take this. And so we went to beagles, and we had this done by White Eagle Laboratory in Pennsylvania. Dr. DeVere is the vet that did the study for us, and the program that we followed was the acute chronic, or the cumulative chronic toxicology study. This is where you give a dose, and then The next time you give a dose in 3 or 4 days, you double that first dose, and in 3 or 4 days, you double that dose, and you keep going up, progressively doubling the dose until the animal succumbs. Once we got to 32 capsules 4 times a day, Dr. DeVere gave up, because all the dogs would do is vomit and had a little watery diarrhea, which I think may be affecting their intestinal flora. Okay? And they ate regular food just like the rest of the dogs did, control dogs, and they gained weight as good as the control dogs. These were young males that were growing. And at the end of the experiment, Dr. DeVere called me and said, Jerry, we can't kill these dogs. I've never seen an anti-cancer substance that wasn't toxic, but you have one. So I was pretty happy about that. And he said, there's no need to kill these dogs and look at their livers for necropsy or anything. He says, I can tell these dogs are perfectly healthy. So we didn't kill any dogs in this study, I'm happy to say. And it proved to us that the Paw Paw material was not toxic and shouldn't be harmful to humans as long as you can vomit. Then the next step was to go to people. And we got some volunteers who were terminal cancer patients, and especially those from Nevada, where you can give terminal cancer patients if the physician and the patient agree, anything experimental. And we did that and got some pretty remarkable results. Dr. James Forsyth at the Reno Cancer Screening and Treatment Center helped us essentially with initial 20 patients, and I have him on tape calling me up, telling me some of the results. Jerry, I have some results that are quite remarkable. This cancer patient, 72 years old, blah, blah, blah. Tumor markers are down, and, they do these tumor antigen markers, and tumor sizes have decreased in just 3 weeks taking Paw Paw. Prostate cancer, similar story. Just amazing. Seen the prostate-specific antigens can be held fairly constant and actually decrease a little bit over a period of several months, which means the tumor is just sitting there. It's not growing, not going anyplace. And with tumors that have metastasized, especially in the neck, for example, we've seen in 3 weeks a reduction of 25% of the tumor mass. With lymphomas, non-Hodgkin's lymphoma, this patient was from Australia actually. She'd been through chemo for 6.5 years, 3 rounds of chemo in that period, then her white cells went crazy and she couldn't control the chemo anymore. So in 35 days with Paw Paw, we got all of her parameters back to the normal levels. And actually I've just had a lady die of lymphoma who was one of our original cancer patients back in 2003. And at that point, when she started taking it in 2003, they thought she'd be dead quite soon. But I know that she was given 3 more years of life with Paw Paw. And she was taking it along with some of the chemo, and it worked quite well for her. But obviously, it's not a cure. But it helped her stay alive much longer. Lung cancer, some pretty good results where people have been bedridden and actually gotten well enough to get out of bed and get around. And we can decrease the development of new tumors, and we can decrease and stop the old tumors from growing. And we can see that on x-rays. And that makes you feel pretty good. Okay? And so with lung cancer, I feel pretty good about that. With melanoma, with lung metastases of melanoma, we have one case where a patient was taking Paw Paw, given essentially just a couple of weeks more to live. And it's been 3 years now, he's still going. And it was metastatic melanoma in the lungs. And he said it hurt to breathe. He said it burned his lungs to breathe. And in 3 or 4 days of Paw Paw, he could breathe again. And it wasn't long, and he's walking. And it wasn't long, he's down to see the cows. And it wasn't long, and he's riding a bicycle. And the last I heard, he was taking care of his sister who is ill. So, in summary then, we've had a lot of patients. Now, I can tell you that the company sells someplace in the neighborhood of 1,500 to over 3,000 bottles of Paw Paw per month, probably averaging 2,000 bottles of Paw Paw per month since March of 2003. And I think just these sales and this use of the product is pretty good testimony to its success. And I feel pretty good thinking that, well, we're helping 2,000 people per month here. I know that it could be more if the product could be promoted, but I think that by word of mouth, the word's getting around, and through websites and speeches. And for example, I'm off to Mexico next week to talk about it again. It'll be my 53rd talk that I've given in the last 3 years on Paw Paw. I think that we're getting the word out, and it's going to continue to be successful, and it may be slow, but I think our progress will be remarkable in the next 10 years, for example. We've seen decreases in tumor antigens, the PSA antigen, the CA2729 antigen in breast cancer, the CA125 antigen. In ovarian cancer and bone cancers, the alkaline phosphatase levels we can knock back down again. Very few side effects, a little nausea and vomiting, maybe about 2%. Not too bad, just cut back on the number. Normally people take 4 capsules per day. If they're heavier people, you can take up to 8 capsules per day. I've had people take more, but vomiting, nausea and vomiting would be the limiting factor. Some people choose to walk themselves up to the nauseating dose and then back off and take that dosage each day. There's probably nothing wrong with that. One person has had itching of our first 90 patients or so. And I've heard of another person since then, but a person took Benadryl in order to get around that problem. No hair loss, no bone marrow depression. Dr. Forsyth tested his patients backwards and forwards for the liver enzymes and the blood, bone marrow depression and all that kind of stuff, and they're clean. The patients are not being given any of the side effects that typically are associated with chemotherapy. And what's another remarkable thing is we've seen really good results with viral diseases and suspected viral diseases like multiple sclerosis. Toenail fungus goes away, for example. Little lipomas, these are little lipid lumps of fat. They're little fatty tumors develop. If you're taking Paw Paw for other types of cancers, those little lipomas go away. In ointment form for acne and athlete's foot, and eczema and psoriasis, for lotions and ointments, although these are not commercially available yet, but someday they will be. That works quite well. One of my chief formulators at the company, I caught her one evening and they're making up some Pau de Arco lotion with Paw Paw. I said, well, how come you're making up more of this? And she says, oh, the kids in the neighborhood are using it. So the teenagers in her neighborhood who have acne were all using this lotion because it was taking care of the acne problems. So I think that that's another use of this product, as well as the pesticide use. And we've got it in a spray, and it's tolerable to plants. In other words, it's not phytotoxic. It doesn't kill plants, so you can spray it on plants. And I've got some pictures I could show you of bean plants, for example, all eaten up by bean leaf beetles, but the row that got sprayed with the Paw Paw, perfect, no problem. And that's right out here in the garden, you know. So it works very well. So I want to thank everybody for their attention.